We are recording. So welcome everyone. I'm so glad that you're all here. I'm really excited to have this discussion today. I real quick want to go around the room if everybody wants to introduce themselves. Just because, like I said, we have a couple of different faces around, which is great. I love bringing all my people together. So yeah, does anybody want to start?
I'll start. I just jumped in before Gabby. Go ahead.
Me? I'll go. So I am a graduate student in Rachel's lab, and most recently, kind of the thing that's been taking up my focus is a genome for the eastern Hawk gibbon. So I'm really interested in this discussion actually because they're also endangered. So I think a lot of this will kind of coincide with that as well.
Well, I'm Kate. I'm also in Rachel's lab. And I work on a couple of different marine genomes. And I'm starting to think about what's next for me. And one of the things I'm interested in is sort of applying the genomic work I'm doing toward, conservation efforts. And I'm thinking about like ways that I can kind of connect the two. So I'm excited about this talk for sure.
I can go. I'm Savannah. I'm also in Rachel's lab. I'm actually working with human, so I'm working on characterizing new regions of the genome, so centromeres and telomeric regions. So I'm not really in the realm of looking at cool new species or trying to help conserve them, but I'm interested in it, so that's why I'm here.
I'll jump in next. I'm Patrick. I'm also a member of Rachel's Weird Genomes crew. I come from the EEB world, and I'm really interested in looking at things at a wider evolutionary scale. So I study wallaby genomes and how they evolved different karyotypes, but they still ended up being a wallaby somehow. And that links together with this pretty well because of the conservation concerns in Australia. So I have a lot invested in this kind of thing.
I'm Emery. I'm also in Rachel's lab. Exciting to say that because I just joined. I'm a first year, so I haven't really found my niche yet, but this is really interesting stuff. So I know I'm gonna enjoy it.
I'll jump in. I'm Becca. I'm not in Rachel's lab. I am in the EEB department, ecology, evolutionary biology. I'm in Eric Schultz's lab. I'm a fifth-year PhD student at this point, so I'm nearing the end of my term. But I'm a fish biologist, mostly the physiological ecology of fish that migrate between salinities. So my particular research is about how the body changes in preparation or response to moving between freshwater and seawater, especially at the juvenile stage, and how that affects population success and success at adulthood.
And I'll jump in. I'm Megan McDaniels, and I am currently a research assistant for a wildlife conservation technology nonprofit called WildMe. But in a past life, I worked for the World Wildlife Fund, which is the world's largest conservation NGO. And I worked on both the oceans and wildlife conservation teams. And I am really excited to share this topic with you all today, especially the genomic perspective. The issue of the Vaquita decline is very close to my heart. It's a great exemplar of so many conservation issues right now. And I think for people who are working at the theoretical level or with human genomics rather than wildlife, there's still a lot to learn from this as a case study as well. So thank you all for having me.
Do you have any other students that haven't introduced themselves yet? Just in case. I'm Rachel O'Neill. Hi. My history is actually evolutionary biology as well, and chromosome biology, and I've been in tangentially related to conservation genetics just because I'm interested in genomes. And because I'm interested in weird genomes, a lot of those species end up being conservation targets. And I think one of the more infamous ones is probably the koala genome. But I've heard about the vaquita my entire life, and I'm very excited to learn more.
And I think, Chris, you're the last one, I believe.
Oops. Okay, I'm Chris. I work as a research assistant in Rachel's lab, and I read the Smithsonian article and I thought it sounded really interesting, and I'd just like to learn.
Me too. And that's how Kate first shared this article with me, the Smithsonian one, and it just started a whole discussion. And I reached out to Megan, and she told me that she worked on in this area with WWF, and so I thought it would be a great place to bring us all together over this kind of topic. So yeah, with that, Megan, if you can share your slides, if you want to try. I couldn't remember how to make that happen.
Yeah, just give me 1 second. I think I should be able to. Oh, it says you still disabled screen sharing. If you want to make me a co-host or the host,
yes,
that can work too.
You are now the
host now. Okay. Okay, great. Can everyone see these slides okay?
Yes.
Awesome. So today I'm sharing with you all the background of the story of the Vaquita conservation crisis before we roll into the more genomic specific discussion based on the paper by Philip Morin and colleagues. And in my work, I've been dedicated to guiding conservation and environmental policy with strong science. And the issue of the critically endangered Vaquita porpoise is really demanding these rapid collaborative solutions from not only researchers, but also local fishing communities in Mexico, legislators, both US, Chinese, and Mexican, law enforcement, and the public. And this recent publication by Dr. Morin and colleagues is a very valuable scenario to consider how we as scientists can use our research as well as emerging genomic techniques to bolster and direct conservation planning, as well as the public perception of conservation issues. And it also brings up questions of where and how genomics comes into urgent conservation issues. A lot of people think about Jurassic Park, recovering the woolly mammoth, and even the northern white rhino artificial breeding program. And that is all well and good, but there is a lot more nuance to how genomics can play a role in conservation and can also help us as researchers build a more nuanced understanding when we go to write about the impact of our work or writing grants, really how much can genomics do to help with conservation. And as mentioned, this was a huge effort, the recovery of the Vaquita porpoise. It spanned 3 countries, Mexico, China, and the US, and involved many, many scientists and policymakers working together. So this is just a handful of them. So I want to start with this quote from the Smithsonian Magazine article that Michelle shared. And to me, when I read this, it was almost like a movie scene, and that makes it very good journalism, but this idea of a desperate rescue, it's the Vaquita's last stand, there's this van rushing to the US border and these bold scientists, and there's drama in the story of the Vaquita, but more than anything, it's a tragedy, and it's entirely preventable. And as genomic techniques are built and discovered and improved, there's this idea of scientists as heroes, and we are, especially when we partner with others, but it's important to always hold that these are, in many of these cases with endangered wildlife, are entirely preventable. So this is an image of the last Yangtze River dolphin, also known as the baiji. And they were the first dolphin species in recent history to go extinct. And this occurred in 2006, and they were fully wiped off the planet due to preventable human activity. And when I say preventable, I mean specific direct causes, less like climate change, but very specific interactions and anthropogenic pressures. And the extinction of the baiji was a national tragedy for China, and it was an international disgrace. And if things continue the way they are now, the vaquita would be the first porpoise species to go extinct in recent history. And likewise, it would be a tragedy for Mexico and the global community, and it too is fully preventable. So what is a vaquita anyways? Many people have never heard of them. They're very, very cute, but often unknown to the public. Their name literally translates in Spanish to the little cow, and they are the smallest cetacean species. And they have these distinct markings around their eyes and mouth that you can see that gives them their nickname of the panda of the sea. And they are very rare, very elusive, even before their population was in the tailspin it is now. And it's very hard to ever photograph them alive. And there are currently no full body photographs of a live vaquita, and the only full body photographs that we have are of deceased individuals. They were first described by scientists in 1958, so somewhat of a recent discovery in the scientific community. And they're one of 4 extant species in their genus. Their closest living relative is the Burmeister porpoise, which is more of an Antarctic species. And in the paper by Morin and colleagues, they are considered the predecessor to the vaquita species. And the vaquita porpoise is found exclusively in the northernmost portion of the Gulf of California. And their habitat encompasses an area just 1,500 square miles, slightly larger than the size of the state of Rhode Island. This is the smallest range of any marine mammal. So although this is an isolated population in a small area, this is not necessarily the biggest risk to their conservation. They're a highly mobile species, and so it's likely that there is very high genetic connectivity in this small population. The Gulf of California at large is one of the most biodiverse marine ecoregions on the planet, and the species there represent 1/3 of all marine mammal species, and there are multiple UNESCO World Heritage Sites located within the Gulf of California. And to name just a few of the many species, there are resident and migratory humpback whales, orca whales, sperm whales, blue whales, sea lions, many species of cephalopods, manta rays, many shark species, marine reptiles like the endangered leatherback sea turtle, and many seabird species. And despite this, the vaquita overlaps with one very, very dangerous animal, and that is the totoaba. The totoaba is a large fish in the drum and croaker family, and they are prized for an organ that they have called their swim bladder. And this is used in Asia for gifting and for traditional Chinese medicine. And pound for pound, the swim bladder is worth more on the black market than gold or cocaine. And this is the crux of the vaquita conservation crisis. The totoaba are highly sought after by local fishermen as part of these networks of organized crime through cartels. to the point that the Totoaba themselves are now considered to be critically endangered. Totoaba are fished using gill nets, which are also known as walls of death because they can spread out for miles across the ocean at different levels of the ocean column, and they will ensnare almost anything that swims into them. So the netting is spaced so that an animal could get its head through, but it can't back out. It'll get snagged on a fish's operculum, their pectoral fins, dorsal fins, as well as flippers and fins of marine reptiles and mammals. And for marine mammals, reptiles, and birds that encounter them, this is often fatal because they're not able to resurface for oxygen. And these nets can spread on for miles at a time even, and they indiscriminately catch Totoaba, which is the goal, but also anything else that swims into them as bycatch. So there's very, very high level of bycatch with this type of fishing gear. It's highly unsustainable. And it is then used both for the illegal fishing of the Totoaba, but also for other fish species in the Gulf of California. And these nets can also become ghost gear, which means this is unintentional nets floating through the ocean. It might be because a storm has loosened them from where they were anchored, or fishermen are just releasing them back into the water. And this happens very frequently with Totoaba poaching, which is done at night so that fishermen are able to do it more secretively, and they will just pull up the nets, cut the Totoaba out, and let the nets go and get out of there. And this is the biggest threat to the Vaquita's survival, the single biggest threat. And because of this, the Vaquita were listed as critically endangered by the IUCN in 1996. Shutting down the trade of the Totoaba swim bladder and removing all gill nets from the Gulf of California is the only way to save Vaquita at this point. But on the bright side, these are completely within human control. It's nothing we can't stop, like runaway climate change, for example. So to give you a sense of the scale of this issue, Totoaba swim bladders are trafficked from Mexico through the US and into China via several hubs. It actually follows the path that many drug trafficking cartels will use for this transnational crime. So it begins in the northern Gulf of California towns of San Felipe and Santa Clara, where the Totoaba are poached and their swim bladders are removed and processed. And this is actually done by a very small group of people. There are 3 Mexican nationals who are involved in cartels that deal with the bulk of this smuggling. So it's actually not, while the poaching can be widespread and rampant, it can be tied to very few individuals. So swim bladders are then moved to border towns like Tijuana and Mexicali, where they are given to Chinese businessmen, also another small group, who coordinates the movement of the swim bladders across the US border and into large airports, especially LAX. And the swim bladders are moved almost exclusively through air routes. And this is almost always on a non-direct flight, almost always packed in checked luggage by Chinese nationals and other smugglers. And they make their way across the Pacific and into several hubs located in East Asia, including Taiwan, Hong Kong, South Korea, and Japan, where they're then moved into mainland China. So because this is an international problem, it has required international solutions. But at every step, corruption and violence has plagued the conservation of the Totoaba and the Vaquita. So in 1992, the Mexican government banned the use of gill nets to capture Totoaba throughout the Gulf of California, but illegal fishing still persisted. In 2005, the Vaquita Refuge was established, and this is the area that you can see outlined in black. And this was decided as the area where 80% of active Vaquita sightings had occurred. So the hope was that it was covering most of the population of Vaquita. but there was never proper enforcement of this area. And as you can see, it's sandwiched between San Felipe and Santa Clara, which are cartel-run towns. And so essentially it became little more than a paper park, which means on paper you can see there's this legal jurisdiction of the area, but it's never been enforced. In 2008, many conservation groups and the Mexican government began to collaborate to develop sustainable alternative fishing gear for fishermen, thinking that if this gear was provided, fishermen would transition to catching seafood more sustainably, and that they could even open markets with these value-added commodities in the US where people could pay a premium for sustainably Gulf of California caught seafood. And there was also a program to buy back gill nets and other unsustainable fishing gear from coastal towns, but it really never caught on, mostly because there's backlash from the cartels seeing this as involvement by outside forces trying to end the trade of totuaba, which it was. And it's very hard to understand the position that these conservationists and local fishermen are placed in. The cartels really terrorize these cities. There's a lot of violence against conservation group. It's been very difficult for WWF Mexico to directly work on this issue. So there's a lot of fear and danger involved with being a part of trying to conserve Vaquita and totuaba. Then in 2015, the Mexican government declared a 2-year ban on the use of gill nets in vaquita habitat. That is the gill net exclusion zone that you can see outlined in red. And this did include enforcement from the Mexican Navy, which was a big deal, but this was still rampant poaching, especially at night, and the poachers were provided with better technology, better intel and equipment than even the Mexican Navy, so it was hard to stand against this. After the 2-year of the temporary ban had expired, the Mexican government actually moved it into a permanent ban, and groups like Sea Shepherd, which is known for combating whaling vessels and such, came into the Gulf of California to help remove gill nets from the water. I'm just seeing I got a chat. Oh, bye, Patrick. We'll send the recording out. And so despite these efforts, vaquita continued to decline. And in 2019, it was considered a 96.5% decline. I'll just leave that figure up for a moment. And you may recognize some of these people. The conservation and policy-making efforts to protect the vaquita, interestingly, included names such as Leonardo DiCaprio, who has a foundation committed to conservation, as well as Carlos Slim on the right, who is a Mexican billionaire and the richest man in Mexico. And between them is former Mexican President Enrique Peña Nieto, and they collaborated on trying to prevent the full extinction of the vaquita. So in 2017, seeing that despite this permanent gill net ban, the vaquita is still declining, the last hope really became this program called the Vaquita Capture, Protection, and Recovery Program, CPR. And the idea was that all wild remaining vaquita would be captured and moved into several floating sea pens where they would be bred in this semi-captivity, and the gill nets would continue to be removed from the Gulf of California, trying to transition fishermen to more sustainable alternatives. And then eventually the original captive vaquita would be released as well as their offspring. And this was a huge operation. It involved both the US and Mexican Navy, international scientists and veterinarians, and even several bottlenose dolphins owned by the US Navy who were trained to help corral and capture the vaquita. It's very interesting. But Porpoise species are considered to be like the hummingbirds of the ocean. They have very fast heart rates and are prone to being stressed. And some porpoise species do well in captivity and some really don't. So it was unknown how the vaquita would do, knowing that they had this naturally high heart rate, and also knowing that regardless, they're just a naturally shy and elusive species. So the CPR program was able to capture 2 adult vaquitas. The first was very quickly showing signs of stress and was released. And the second was a breeding age female, and she was doing okay for a while. People were really, really hopeful. They were constantly monitoring her vitals. But after several hours, she suddenly took a sharp turn for the worse and was showing signs of severe stress. And veterinarians were trying to stabilize her and get ready to release her, but she suffered from cardiac arrest and they were not able to revive her. She ended up passing away. And this was a huge loss for the species, being a breeding age female who could have helped produce more future vaquita, and a huge devastating blow to the program. And the program was shut down after this. And NOAA representative Barbara Taylor said that even the loss of 1 vaquita was too much, and that was the absolute threshold of risk that this program was willing to take. So slightly on the bright side, the tissue samples that were used for Dr. Morin and colleagues' publication that Michelle shared came from this adult female. So she was able to contribute to the development of the genome, but sadly passed away. And it's now believed that the population of vaquita in 2021 are in the single digits. Conditions are very bad in coastal fishing communities in the northern part of the Gulf of California. These areas also rely on tourism and sport fishing, and that has been lacking due to COVID. But there is still hope for the species. This is an image in 2019 of an adult female vaquita with a calf, and this is despite all of the gill nets in the water, despite very intense fishing pressure for Totoaba. These vaquitas seem to be able to breed well. They're finding each other even though there's fewer and fewer. And females are able to produce a new calf about every 2 years. So there is still a chance. This gave many scientists a lot of hope that even though the CPR program had been considered a failure, they are still able to breed in the wild on their own. And this is where genomics really comes into the story, because the question now is, can vaquita genetically recover from the extreme bottleneck they're experiencing? And has their population ever been this low in the past? And political will, especially in Mexico, and somewhat understandably, has been declining. And there are many who don't even want to take action on banning and removing gill nets. And this has been an excuse to continue the status quo, saying that the vaquita are doomed to extinction regardless because their numbers are so low. And people have been wondering if inbreeding depression would be so high from being in the single digits of population, that is this inevitable no matter what actions are taking. Scientists did infer in 1997 and 2005 that vaquita's low heterozygosity was due to long-term small population size and not the recent bottleneck, which was a good sign. But it was really this study that just came out from Dr. Morin and colleagues that found that vaquita are tolerant of small population size and the genetic consequences of bottlenecks, and that they have a good chance of persisting as long as the gill nets are removed from their habitat. And this is really important because it is completely reframing the issue as one of there is still a chance, and has also underscored the urgency but the potential for the recovery of the vaquita porpoise. And I'm going to turn it over to Michelle now, unless anyone has specific questions right now. Otherwise, you can save them for a discussion at the end, but I'm going to turn it over to Michelle to share more about Dr. Morin's publication specifically.
Hey, can I ask a question about the conservation effort?
Yes, please.
So you pointed out it was notable that when they expanded the gill net exclusion region, that they got the the Mexican Navy involved. And I'm just curious how they
Mhm.
achieved that, how they were able to achieve that, given the political climate as well as, you know, illegal activity that was going on in that area at the time. It seemed
Sure.
like quite a
So there
coup.
was a lot, yes, there's a lot of international pressure around this issue from institutions like CITES, which manages the illegal trafficking of wildlife. So I had mentioned that there were several UNESCO World Heritage Sites in the Gulf of California. UNESCO was considering these to be on the list of threatened heritage sites, which is, I think, a big blow to Mexico and the draw of that area. But probably one of the biggest drivers of this action was the US sanctioning seafood from this area of the Gulf of California. They produce a lot of shrimp, a lot of corvina, I believe it's called, which is a smaller relative of the totuaba. And the NOAA and other federal agencies were threatening a sanction against Mexican seafood to put some pressure on this issue. And I believe that there currently is a sanction against the seafood. I know it all comes down to the money sometimes. But yes, and it's the vaquita directly, but it's also all of these other species that come to the area that are being killed through this process. Thank you, Dr. O'Neill. Okay, turning it over to you, Michelle.
Great. Thank you so much, Megan. That was great background. Kate, you unmuted for a second. Did you want to say something?
Yeah, we could talk about it after if you want, or I can ask now, whatever.
Go ahead, ask now.
Well, I was curious about the totuaba. Because it almost seems, well, that's like the crux of the problem, right? So like, is there any effort toward like aquaculture with the totuaba to try to reduce the natural, fishing pressure?
That's a great question. They were actually approved to have aquaculture for totuaba, and some of the fish are released into the Gulf of California to help with that wild population. This is a kind of a perennial debate around these illegally traded wildlife parts. For example, rhinoceros horn. There has been some questioning around, could we make synthetic rhino horn, or could we take farmed rhino like in South Africa, who frequently cut off their horns to deter poaching? Could we start to sell that? But it's been found through several studies, some conducted by the World Wildlife Fund, that this actually is just fueling that demand. So this demand for totuaba is due to an unproven belief that there's health benefits, also the sense of it's rare, expensive good that you can gift and get goodwill from your boss, say. And in continuing this demand or having some in the market, it's opening up that demand to still happen for wild individuals. It's very hard to tell what's wild caught, what came from aquaculture, what is synthetic. And I think that overall the best thing that people are hoping now on the Chinese side, cracking down on that trade as well as trying to reduce the demand. But aquaculture has still continued to help restock wild individuals. Totuaba are very interesting. They're very, very imperiled because of overconsumption of water in the, I believe, Colorado River in the US. And for many years, the Colorado River never fully emptied into the Gulf of California like it used to, because so much water was being drawn along the way, and that estuary areas where totuaba breed. So that's another big pressure on their population.
Cool.
Yeah, I'll just jump in here.
That
actually like segues into a question that I had. I was wondering too about. I know that they're very aquaculturable as a fish, but I was wondering about like the crosstalk between conservation efforts for totuaba and for vaquita, because so you answered a question that I had about a CITES sanction that meant that even though they are aquaculturable, they now can't be traded internationally, which now that makes sense based on your comments about, they just want to cut off the supply. But,
right,
in terms of the gill netting for totuaba is illegal.
It is.
And they're actually listed as a bycatch species if they are caught by a certified commercial fishery. So I'm wondering like if there is crosstalk in those conservation efforts, because it seems like there would be more backing if that were tackled as a team.
Crosstalk between the fishing industry and conservation?
Or just conservation efforts for Totoaba and also for Vaquita, at least in this one area where they coincide.
I see. So vaquita have often been used as what's considered an umbrella species or flagship species. So in protecting the vaquita, you're also protecting Totoaba. So the efforts that help to conserve vaquita also help to conserve Totoaba. They're just cuter than Totoaba, which helps, and more critically endangered. So I think that very often these discussions and planning includes all different people from different backgrounds, including industry. And Conapesca, which is the Mexican version of NOAA, essentially, has always been very involved in the conservation of vaquita. And I forgot to mention, Kate, another draw for if there were to continue to be demand for the Totoaba swim bladders is that often in the traditional Chinese medicine, wild caught is considered superior to farmed, which you see with lion bones, tiger bones, rhino horn. It's just seen as having stronger properties than captive raised or bred individuals. So Becca, I don't know if that answers your question, but
yeah, there
is a lot of overlap.
Yeah, it's an interesting amount of overlap, and I guess my underlying question is, what is the actual fishery that they are bycatch of? Because I couldn't find that anywhere. Granted, I didn't dig very hard, but even for the Totoaba information like that NOAA gives, for example, they're not actually, I should check, maybe Sea-Watch would have it, but they don't actually list what they're a bycatch of, and so I'm wondering about the regulation of those target species. Like, what is the target species that's actually
target species would be target species would be shrimp and corvina mostly, and it was an iterative process, the banning of gill nets. So initially it was banning of gill nets to catch Totoaba, then it had to become banning the catch of Totoaba, period, and then it had to become banning of gill nets entirely, because people would say, oh, I'm using these to catch corvina, but really it was just a way that they could go out at night and catch Totoaba. But the poaching itself typically is directly aimed at catching Totoaba, and then if they catch a sea turtle, if they catch a shark, if they catch a vaquita, it's bycatch.
Yeah, I think what's interesting, it's interesting from like a fish biology point of view, right? Because it feels like the story is being pinned on Totoaba, when in reality it's like a shrimp's fault, right?
It did initially, yes, it initially began, so in China there was a large native fish species that I didn't mention that has nearly gone extinct because it was being hunted for its swim bladder for traditional Chinese medicine. So it was only in the last couple decades really that Totoaba became this prize for illegal trafficking. And initially it was shrimp, corvina, other fish species in the northern Gulf of California that were resulting in the bycatch of vaquita. And so it's like these larger industries that are somewhat easier to regulate, especially when they're in EEZ zones for a country. It kind of, that pressure had been eased, but then right after it came this new pressure and demand for Totoaba.
Right. Yeah, no, it makes sense. I think just the decision about how to frame it is interesting to me, that it's framed as like Totoaba are doing this, whereas in reality it's poachers and commercial fisheries that need some gear management action are causing this, not the Totoaba.
Right, not the Totoaba directly. So
yeah, no, it's just interesting to me.
Yeah. Anyone else have any questions, or do you want to bring up the paper, Michelle?
Yeah, I'm interested in bringing this to a larger discussion. I don't want to like dive deep into the paper on my own here, but just to highlight for anybody that didn't get a chance to read it, they were able to produce a really high quality genome thanks to the live cells from the unfortunately now past captive animal. With this, they were able to confirm that the heterozygosity of the genome is very low, and it is thought that they have pretty much bred out potentially harmful mutations. And also they did some evolutionary backtracking to see what it seemed like the populations looked like based on the genome. And basically it seems like they've been small for a long period of time, as Megan indicated. And so that the idea from this paper is that it has encouraged like the Mexican government or other groups to continue conservation efforts for the vaquita because it is savable, is the idea. But now I want to bring this into a larger discussion. Oh, beautiful. Oh my God, it's a vaquita. So yeah, we can just start off with the first question here, which is how can or has the vaquita, or genetics or genomics, contribute to the future of vaquita recovery? And this is kind of an open-ended question. If anybody has any thoughts about like what's possible to do, is there any way that we could use genetics or genomics to do something to help the population? Open-ended thought experiment, if anybody has any thoughts. Rachel,
I wanted to take a step back because it's related to this exact question, and pose the question for the group, sort of a two-level question, right? So one is how, and I did read the paper quickly today, and it was so the first thing I'll say for you people doing genome assemblies, this is a beautiful genome. And one of the reasons it was so easy to do, well, there were two reasons. One is the San Diego Frozen Zoo, which Ollie Ryder runs. And his goal is to make, and has been for decades, and I've worked with him on many projects. He's the group that helped with the cross-species cloning of the Przewalski's horse. So his goal is to make cell lines from every species on the planet. Now, one of the reasons this genome assembly was so beautiful is because of the unexpected finding that it is not very heterozygous, right? So that's the reason that diploid genomes are so shitty. But the question I have is that I saw these major dips in population size, effective population size, and there were multiple. And so I'm wondering, how the authors were able to take the leap that, oh, it's not a very zygous population, it's a very homozygote population. The effective population size has dropped many times, so it can withstand these population crashes, versus this is why they have all these population crashes. And these population crashes are so tenuous that any external impact, and I noticed they talked about, the last glacier times, etc. So obviously fishing would be a small but significant external impact if your population sizes are constantly dipping like this. And so I was wondering how the authors, made that distinction, if anybody had any insight into that. And I don't have an answer.
I also don't have an answer for that, but what I will say is I'll just comment on, like, I do think it's really interesting to think about the stochasticity of populations, and I think it's good evidence for, I think whenever you're dealing with like an endangered species and population fluctuations, like, most people don't understand that there's quite a lot of stochasticity that goes into populations, and particularly in marine species, actually, they tend to have a lot of stochasticity because they're so tied to this like aqueous environment that they live in. And it's interesting to me to see evidence for the fact that, like, yeah, this is a species that maybe naturally has a lot of stochasticity and can be fairly robust against that because, right, of course, because managers are going to be looking for a return on investment. And if they feel like they're going to invest money into a population that's going to come back but not be very good, or only going to come back and then not be sustainable into the future, that's like not worth it for them, you know. And that's these are big arguments when you think about things like the Florida panther, right, with the kink in their tail. Like, there's a lot of argument about why, like, is it even worth it to bring them back, because they're not, you know, they have low genetic diversity and are dealing with some of these repercussions. But for a species like this, where we can show that it might actually be worth it, that's pretty cool.
I agree. I thought it was very interesting in the paper, several other species that they included, like orca whales and brown hyenas, that naturally have this long-term low heterozygosity, and considering this compared to species like the northern white rhino, that such a huge effort is being invested to recover them, and wondering what that return on investment is. And that kind of brings me to the fourth question on here. What does a world without vaquita look like, and what are the costs and benefits of protecting them? And I would love to hear what people think about that for vaquita specifically, other species that are critically endangered, that huge amounts of money are going into protecting them compared to other species that are vulnerable or endangered or near threatened, and how we use genomics to justify whether we'll make that investment.
Well, I think, you know, to sort of follow up on both of those points, I think one of the really interesting points is that, and tragic as well, you know, when I read it in the paper and then you told me again, I had little inside tears, is that they can't be held in captivity, right? So really demonstrating that you have a population that can be sustained over, you know, many millennia, with very low effective population size, and sometimes very small populations, I think does contribute to the significance of being able to try to control external factors that impact this population. Because I think you're right, you can justify that there is a chance, and it's not like the white rhino where we can, you know, put them in zoos and try to breed them and do breeding programs to get more outbreeding and more increased genetic diversity, because this just may not be possible, and it may not be necessary. So in some ways, I think that this genomics data actually supports the conservation efforts and the cost benefits of protecting them. But that's my personal opinion.
Yeah, and I'll say on the in terms of the conservation efforts and things, you know, I don't want to come off as too negative. I'll just sort of preempt my comments with that, because as a fish person, and obviously this has probably come out, that, like, you know, it frustrates me a little bit sometimes to see a mammal, a charismatic mammal, being an umbrella species, and because I genuinely don't believe it always works. I think we have evidence to say that when we use umbrella species, sometimes it can work, but if you're using an umbrella species to cover the species that is actually the problem, or whose targeting is actually the problem, it's like we're putting a band-aid, and that's what makes me nervous about these practices, right? Like, if we want to save the vaquita, if we only focus on that species and try to use it as an umbrella, it's like we're putting a band-aid on the vaquita problems, right? When really the problem is that Totoaba are being illegally caught and sold. And yes, there are regulations and conservation efforts going toward them, but if all the money and attention is going towards the vaquita, that's money and attention that's being taken away or turned away from the Totoaba, right? When again, if we just turn those heads, and we could still use vaquita to convince people to turn their heads, but if we were, you know, instead of blaming the Totoaba for the issue, if we told people, like, Totoaba are being
targeted, and because of that, the Vaquita are getting hit too. So turn their heads toward the Totoaba and say, look, if we spend some money on just shutting this down, it'll save both of them. And again, like instead of making this sort of bad guy situation for the fish, we kind of partner them together and we say they're friends, and why don't we save them both and try to be a little bit more strategic with our efforts. And I know that's hard because I know that everyone wants to protect the cute thing, but I think fish are kind of cute. So
I actually had the same thought as you though, and I like totally agree. And I just looked quickly. I was like, oh, I wonder what like genomic or genetic information there is on Totoaba, and there was like no genome, not that much info. Like, so it's an interesting idea, and I completely agree.
yeah, and again, I'm like not trying to be super negative about what's going on, because I think like I'm very pro conservation. I think we should be doing this. I think the thing is like when you Google Totoaba, like the first couple articles that are going to come up are like Totoaba are killing Vaquita, right? And that's the framing issue.
It's also the idea that these ecological roles that both the Vaquita and Totoaba play, and this world without Vaquita as well as Totoaba, maybe from an ecological perspective, it wouldn't be that different than what the Northern Gulf of California is like now. And perhaps it's better to choose a species that is more of a keystone to that environment or has a bigger impact, because I don't know if there is currently, I don't believe so, but there was previously sport fishing of Totoaba. So at least there was that economic incentive to protect them. For Vaquita, they are so hard to ever see that there's no tourism around them. And both species are limited to the northern part of the Gulf of California. So in terms of an umbrella species, the entire Gulf of California is very biodiverse, but picking only a species at the very top is the top portion of that geographic range is perhaps not serving the best as an umbrella species.
How much of this is not just using it as an umbrella species, but using it as a tool to tap into, you know, the empathetic nerves that in education, right? So I would be surprised to see Leo standing there with a really cute picture of a fish with a billionaire and the president of Mexico, right? So but the added benefit of this is that there are other species that are being, you know, that conservation efforts can start to emerge, and we can actually start to recognize, oh, this is actually an entire ecosystem. This is how this ecosystem works. This is how this one major human activity is affecting all of these things, and this is how to change it, right? So, you know, in a way it's an umbrella species, but it's also an educational tool. And sadly, it is, you know, it is the cute mammal. And people don't.
I love the charismatic megafauna.
Yeah, right. And it's unfortunate that that's the way we have to teach conservation and educate the population. But, you know, the fact of the matter is, if you were to say somebody takes this fish and takes their, you know, bladder and takes it over to China and sells it, you're going to get a lot of people who are like, so what? And that's unfortunate. And I think it's a way to start the conversation of, this is why. Oh, and let me also remind you that there are other species like fish that are impacted, or, you know, shrimp or whatever may be impacted in that entire ecosystem.
And that's like one of the most, you know, that's like one of the biggest challenges of conservation is like, why does it matter? Or species extinction is the why does it matter?
I think it's a great way to show, though, it's not, I don't see it as an umbrella species, but more of a sentinel, right? So as a species that indicates we have an ecosystem that we are now, you know, dramatically impacting. This is one obvious way. And so it's more of a, you know, there's a big systemic problem, and maybe thinking of conservation in terms of sentinel species that can indicate, you know, or draw attention to, or, you know, if money is being brought in, there's an added benefit.
Yeah.
Oh, Go
No,
ahead,
sorry.
sorry.
I have a quick question actually, I'm gonna knock us off the conservation track just for a second because I'm just curious. Given that it's such a stochastic species, and I couldn't find this actually in the literature anywhere, but I'm wondering where we got the range estimation from. Like, is there a paper or a study that was done? Because I couldn't find any primary source on how they've decided that.
Do you mean like, oh gosh, this range estimate? Yes, I believe that's done through acoustic monitoring. That might be something that's less publicized because it's more of the technical fieldwork aspect. And I can try to find some follow-ups to share with you all outlining this better, but vaquita are mostly surveyed through acoustic monitoring as well as some visual censusing. So this, I can't see the bottom of my screen right now, but I believe it was NOAA who conducted this. And CERVA, who is a consortium of international and Mexican researchers. But yeah, it's mostly done through acoustic and visual surveys.
Yeah, just because the ecologist in me is always curious about distribution data and like where it's come from, and also like, you know, how that acoustic monitoring data aligns with the gill netting, the location of gill net catches, if that's something.
Right, I believe that the Totoabas range is a bit larger than the vaquitas, and there is a specific area, the estuary area, where the Colorado River opens into the Gulf of California that Totoaba will breed in this, or their spawning season, and that is really when there's targeted efforts to fish for the vaquita or the Totoaba, and I believe that is in winter typically, so it's a pretty tightly overlapping area between where those gill nets are and where the distribution of the vaquita is.
And do they know where the vaquita's reproductive grounds? I'm a fish biologist. Like,
I'm not sure that
are they just kind of like, I guess my big question is, you know, like, are they just distributed throughout, or do they have hotspots?
They are fairly distributed throughout. They're very hard to find again. So I think that census type data is the best that there is, and no one really knows if there's hotspots, but I think that they're very mobile and they're found pretty much throughout this range area.
I do want to acknowledge real quick that it is 3:30, so if anybody needs to go, that's okay, but I'm happy to keep this conversation going. So yeah, just gonna put that out there.
Do you want to pick another question, Michelle?
Sure, unless Kate, did you have something you wanted to say?
Unless anyone else has a
I
follow-up.
was just going to say, from like a genetic standpoint, I wish that they were easier to find, because with so few, it would be awesome to just look at, you know, the genomes of all the individuals and see what kind of variation there actually is. Because, you know, obviously with one genome, you're just looking at one individual. But I don't know, it would be really interesting to see at a little bit wider scale, like what's going on there, so you can make better decisions about what conservation measures we should use.
Need to hire more of the trained US Navy dolphins for that.
Yeah.
I was actually going to ask that before we switch. I have two questions related to that. One is, do they have natural predators, and are other, you know, dolphin, sharks, you know, because they're kind of small, right? They may be fast, but they're relatively small. So I don't know if they know any of this. But,
yes, there is evidence that there is predation of vaquita by different shark species, and that is from parts of vaquitas found in shark stomachs, and also what do you call it? Premortem bites by sharks on vaquita bodies that have been found.
And so the other question I had was, the other species that are in the genus, how closely related are they? And is there any genomic information or efforts to look at genomic information on those? Because that sort of comparison of heterozygosity, post-divergence, and reproductive isolation regions and where you've got recombination hotspots could actually inform the species and how to set up conservation efforts.
Yeah, so okay, I'm unmuted. There are 3 other species in their genus. The closest related is the Burmeister porpoise, but I don't know, or I don't remember if this paper that we're discussing included the relatedness between the 2 species, but there are many, many more of those porpoises. I would assume higher heterozygosity, and I don't believe there's been efforts to sequence their genome yet, but it would be very interesting to have a follow-up study with that.
Yeah, I just looked, there's only a mitochondrial genome.
I'm guessing it's on the VGP list.
Maybe,
I have no idea.
That paper does mention as well that those other genuses are just located elsewhere, so there's not like population intermingling, which I learned from the GAP conference on Tuesday is much more common than we would think. It's that kind of hybridization, but yeah, it seems like the focus has been on Vaquita, probably because of how endangered they are.
Yeah, and they're one of the warmest water porpoise species, and there's no real viable surrogate for them if they were to be considered for the artificial breeding route.
And are they more of a pod or social species, or are they more?
No, they are not social. They have been seen occasionally in pairs, but it's believed that they only come together really to breed. So they're pretty much solo.
Antisocial.
Introverted.
Yeah, that makes it so much harder, right?
A lot is stacked against the Vaquita. Besides their fairly productive breeding level, when they're able to find each other, a lot of other things are stacked against them. I would love to ask, just while everyone's still here, I would love to ask the third question about the role of geneticists in educating and collaborating with conservation decision makers and managers, as well as the public, to increase like a genetic literacy and understanding of what options are available, especially before species become critically endangered like the Vaquita. And I would love to direct that question to anyone we haven't heard from who feels comfortable sharing.
I'm thinking.
Take your time.
I think maybe in the meantime, Megan, you could talk about what you told me recently about how you became interested in genetics as a tool, and how maybe conservationists aren't always aware that that's a possibility.
Sure. So for a long time, we've all been probably involved or aware of the importance of STEM education with the public and with youth especially, and that is hugely important for more equitable research, for more diversity in STEM, as well as for more support for issues like conservation. But more and more, I think there's been this question of how scientists can better collaborate with each other. And so not just the impressionable youth, but also those conservation managers and policymakers, and what is the role of researchers in communicating with them. And personally, I had been very interested in applied research and population ecology or conservation, but in the past year, I was working at an ecology and evolutionary biology center in northern Kenya, and I helped to manage the most high-tech genomics lab in the African continent outside of South Africa. And it was amazing to learn about the capability of this lab and how many applications it could have to common species, to critically endangered species, as well as to livestock and the overlap between wildlife and livestock in that One Health perspective. And it wasn't until I was able to be side by side with geneticists and to learn about how these tools and techniques work that I saw, oh, it could be incorporated this way or that way, or answer that question, or helped me and others to form these questions we never even thought to ask. And we had a bit of an issue of the uptake of this lab initially, because this was supposed to be a resource for Kenya and East Africa. And we had, I think, this idea that people would come flocking to it, but it wasn't enough to just offer this in isolation. It took many conversations and discussions with people who managed endangered colobus monkeys on the coast, or certain species of drought-resistant cattle, or breeds, excuse me, of drought-resistant cattle, before people were able to start to see how these tools could help them in their work. So I would love to hear from you all as budding geneticists and full-fledged geneticists and researchers on how those collaborations can be made, and even while you all are still in the PhD process, how you can start to have inroads locally or learn those skills of working with other stakeholders.
I feel like I don't know, there has to be some way to, and maybe there already is, to like, I don't know, bring everyone together in one place. Because I feel like people might just not realize the capabilities of like merging all these fields and to make people more aware. But it's trying to figure out the best way to like get that information out there the most efficiently, which I'm not sure what the best method to do that would be.
I think it would be interesting, the GAP conference, or Dovetail Genomics conference that Michelle mentioned, learning how they did outreach to conservation practitioners and invited them, or publicized this event to conservation practitioners. It would be really interesting to see if that was considered, and if so, like picking their brain how they went about doing that, because at so many levels, like having redundancies in incorporating different groups in this work would be interesting to learn about.
So one of the things I want to remind everybody about is the emerging Earth Biogenomes project. And its goal is to sequence all species of everything, agnostic to how cute it might be. But its governing structure requires participation and input, not just from local governments, but communities. And I think that is also one of the biggest challenges in conservation, is not just, it's one thing for us all to be like, oh, let's do some genomics on some really cool endangered species, and we're seeing this with, things like the koala or the kiwi, where the indigenous peoples are like, actually, this species is very important to our culture, and, they need to be participant in those conversations. And I think Earth Biogenomes Project has a very good outline for how to do that. And I think that literacy and that buy-in is critical in conservation.
Yeah, I'll say in my experience, in terms of collaborative efforts, the most successful collaborative efforts that I've seen and or been a part of have been those that honestly began at conferences. That's a great place to make connections, and I've seen it done just, you attend each other's talks and you realize you have a common interest, or I've actually seen it targeted too, where a conservation-based organization will give a talk and it will actually end with a call for engagement, and ask for scientists to buy in and then have meetings. And then beyond so that's kind of like the seeking part of it, right? But then you actually have to have the collaborative portion. And I think that's where a lot of times, sometimes these groups miss the mark, in that like you make connections, you think that's great, we've met, we've talked, but it really takes iterative meetings to get the most out of those relationships. And so working groups are the most successful that I've seen in terms of connecting scientists and conservationists is that you meet, you know, like once a quarter or something like that, and talk and give updates and figure out where you're all at. And then in terms of actually reporting that out and engaging with the public, I honestly believe that there's no one size fits all. I don't think that there is an answer to that. And I think what it really is, it has the same issue as any SciComm project, which is you have to think about your audience. And that's going to change based on the species that you're working with, based on the community, the breadth of your audience, the age of your audience. And sometimes genetics are going to matter, and it might convince some people and really get them excited and on board. And sometimes they're just, sometimes that's going to be beyond the scope of your audience, and they're not going to care. And they're not going to care because it's not important, but they're not going to care just because they might not get it, right? And so again, like, I think it can be really, the genetics and genomics portion of this can be really important in engaging those audiences and getting them really excited about stuff if they have the scope to understand it and to appreciate it. And again, that's going to be based on which audience you're dealing with.
So I think that understanding and appreciating is like one of the key things that I'm a big fan of, right, is genetic literacy. And how important that is, and how little of that there is.
Rachel has encouraged that through her class that she teaches. But sorry, Emma, you were going to say something?
Yeah, so actually, before I was interested in genomics, back when I lived in Florida, I would volunteer at the zoo, and we would do educational like programs to teach kids about the bottleneck effect and everything that's affecting the Florida panther. So it's interesting looking at it now as a geneticist. Back then, or excuse me, now, and back then, I was more looking at you know, economics and conservation, and I was like, I don't understand, you know, genetics, and so I can see the added difficulty now of the layman terms and getting people excited about genetics when, you know, they're like, oh, it's a cute panther. Well, I don't care about genetics.
Well, and that's like to me, that's what's frustrating about conservation work. And I feel fortunate in that the species that I work with is a species of concern, alewife, and I feel fortunate in that, like, my argument is always, well, there's an economic reason to protect them, as there is with many, not all, but many fisheries. But when you have these species that, you know, don't necessarily have a direct economic tie, there's always this, like, there's always going to be a group of people that asks, like, well, why should I care? Like, it's cute, but so what, you know? And to me, that's like, I mean, obviously we care and we have reasons, but communicating that to other people is challenging. And I think that's where, like, the genetic genomics component could be really convincing to people who are going to take a more practical stance.
Yeah, I think particularly when working with the communities who are directly living with these species and see these species as money, as school fees, as housing, as food, and it can be really, really hard to make that case, and almost a little ignorant of us as researchers to say that this should be valued because it represents a distinct species. But if people aren't able to eat or afford to care for their kids, it doesn't matter. So definitely making the economic cause sometimes, I mean, very often supersedes almost any other cause, for better or worse.
Well, this has been awesome. Thank you so much for facilitating, Megan. And I'm so glad that you all were able to make it and join us for this discussion. I'm going to go ahead and stop the recording.